Hello everyone, let's get started. In today's video, we're going to be talking about the different types of culture media, including a type of media called the special media. The type of culture media that we use in the microbiology lab can be solid, semi-solid, or a liquid or broth. We can add ingredients to water, for example, to create a broth. And we can add ingredients and agar to water to create a solid medium, the type that you would find in an agar plate. We can also take a broth medium and we can turn it into a semi-solid medium by adding a little bit of plain agar to it. Over here on the left-hand side of the slide, you can see a container of powdered agar that was purchased from a supply house. This powdered agar is harvested from a certain kind of algae that use agar in their cell wall. Remember, agar is just a complex carbohydrate. What makes it such a nice medium is the fact that it stays solid at a range of temperatures, including the high temperatures that we sometimes use in the incubator. And it's not digestible. In other words, most microbes, the vast majority of microbes, cannot digest agar as a nutrient. They'll grow on its surface, but they're unable to break it down and use it for nutrition. There are many, many different types of culture media that are used in the microbiology lab. They allow for us to culture all different types of microbes, and each type of media has its own unique recipe. Some media will allow virtually any microbe to grow. These are the types of media that we often refer to as growth media. There are some types of media that will encourage just bacteria to grow. There are other types of media that are designed specifically for fungi. Some media will only allow non-fastidious microbes to grow, while others are going to support the growth of fastidious microbes. Now, these terms are new for us. A non-fastidious microbe is a microbe that is relatively easy to grow in the laboratory. In other words, it's a type of microbe that has very common nutritional requirements and very common conditions for growth. In comparison, a fastidious microbe is one that's very difficult to grow in the laboratory, usually because they have very particular nutritional requirements or they have very unusual conditions that are required for growth. So we have media that will grow most non-fastidious organisms, but we also have media that will help us grow fastidious microbes. Still other culture media are designed to support the growth of one type of microbes, say bacteria, while inhibiting other types of microbes from growing. Some actually contain ingredients, typically indicator dyes, that will allow us to look at a culture container and know immediately whether or not the microbe that's growing is able to perform a particular metabolic reaction. That can be very helpful to us when we're trying to differentiate between very closely related types of bacteria. On this slide, you're looking at some very common culture containers. Over here on the left, we can see an agar plate. The agar plate has agar poured into it. Remember, we mix up a powdered agar with water in the laboratory, and we can add all kinds of other ingredients to that mix, depending on the type of media that we're trying to make. We then heat that up until the agar melts, and we pour it into the plate. Once it's solidified, the agar will stay solid at both cooler temperatures like room temperature, all the way up to the higher temperatures that we use for incubation. In the middle, you can see agar that's been poured into a test tube. This is referred to as an agar deep. Now, you would not use an agar deep the way you use an agar plate. An agar plate has lots and lots of surface area for growing bacteria. But the agar in the tube has only a very small surface area at the top. Instead, we use an agar deep to grow organisms that have different relationships with oxygen. And we do that by using what's called an inoculating wire instead of an inoculating loop. We take the wire, we put some bacterial cells on it, and then we stab the wire down into the agar. What that's going to do is allow organisms to grow deep in the agar that might have trouble growing in the presence of oxygen. Some oxygen can actually penetrate agar, but it can only penetrate a short depth. So if we're trying to culture bacteria that have a bad relationship with oxygen, either they don't use oxygen or they find oxygen toxic, they can actually grow in the deeper reaches of the agar where they're going to be protected from oxygen exposure. The tube in the middle here is just a broth tube. As you can see, the broth is much more transparent than agar is. And finally, on the right-hand side, we have an agar slant. When an agar slant is prepared, the tube is placed on its side while the agar is hardening, so that we end up with this slanted sort of shelf of agar surface for growing bacteria. We can spread cells all along this slanted edge and grow them inside an agar slant. Now, of all of the many hundreds of different types of culture media available to us in microbiology, we can break them down into 6 large groups. The first 2 groups are considered growth media. These include basal media and enriched media. Basal media is a type of culture media, a type of growth media, that will support the growth of most non-fastidious bacteria. Basal media has a short list of ingredients in it, and it meets the nutritional needs of most non-fastidious organisms. Basal media is also sometimes referred to as chemically defined media because we know exactly everything that goes into the medium. Nutrient broth and nutrient agar are great examples of basal media. Now, enriched media is quite different. Enriched media actually supports the growth of most fastidious bacteria. And that's because enriched media has what's called a complex ingredient added into it. Complex ingredients include things like blood or serum or egg yolk, even sometimes things like meat digests and milk can go into enriched media. Enriched media, in other words, contain a complex ingredient that is very nutritionally dense. Blood, for example, contains many different individual molecules, proteins and fats and carbohydrates, that just might meet those very special nutritional requirements that fastidious bacteria have. We sometimes call enriched media complex media because it has those complex ingredients in it. We also sometimes call it chemically undefined media because these complex ingredients contain so many different molecules that we can't just list them the way we could with a basal medium. A good example of an enriched medium is called blood agar. Blood agar is made by preparing a basal medium and adding some type of animal blood to it. On this slide, on the left-hand side, you can see a blood agar plate up at the top. Blood agar is prepared by making a basal medium and adding blood to it. We often use blood from sheep for this purpose. You can see that the agar is very deeply red, often a bright red color, and it will support the growth of many different types of fastidious bacteria. Down below it, we can see another kind of enriched media. This is called chocolate agar. Now, unfortunately, it does not contain chocolate as its complex ingredient. Instead, it's another recipe that includes blood. But with chocolate agar, the blood has been cooked before it's added in. It's been heated up and cooked so that some of the molecules in the blood start to break apart. There are some fastidious bacteria that prefer chocolate agar to regular blood agar. Over here on the right-hand side of the slide, we can see two broth tubes. I put this in here to show you that broth tubes can be helpful for us when we're growing cells in the laboratory, because broth tubes begin as a very transparent type of media. If you were to hold a piece of paper behind this tube with writing on it, you'd be able to read the writing through the broth. But after you inoculate broth and incubate it, this happens. The broth becomes cloudy or turbid, as we say. This turbidity tells us that we've been successful in growing cells in here. Now, the reason we might choose to grow cells in broth instead of on an agar plate is because in a broth culture, we're going to be able to keep the cells separate from one another. We often put these broth tubes onto a shaker device in the laboratory, a device that will gently swirl the broth as the cells incubate and grow. We can then use broth to inoculate other culture containers, including agar plates. So both basal media and enriched media are referred to as growth media. The next two types of media that we'll discuss are called special media, and these include selective and differential. Selective media is used to select for or support the growth of certain species of bacteria, while selecting against or inhibiting the growth of other species of bacteria. In other words, this type of media selects for a particular type of bacteria that we want to grow, while inhibiting all the other kinds of bacteria. It allows us to pull out certain species of bacteria from samples that might contain mixed bacterial populations. Differential media allows us to differentiate between closely related species or strains of bacteria. Remember, when we talk about strains of bacteria, it's very similar to breeds of dogs. They all belong to the same genus and species, but they have some slight genetic differences that make them different enough that they're their own strain of that particular species. Now, differential media often contain those indicator dyes that I mentioned earlier, dyes that will change color under different conditions of pH. So when we grow these closely related species, we can tell one from the other based upon whether they perform certain metabolic reactions. The organisms that do perform those metabolic reactions produce byproducts that alter pH in the medium and cause the indicator dyes to change color. Let's first talk a little bit more about selective media, and we'll use the example of mannitol salt agar, or MSA. Now, mannitol salt agar, like the name suggests, contains a relatively large amount of salt, particularly sodium chloride. Most bacteria don't grow in that much salt. If you think historically, sodium chloride has been used for many years as a preservative for food. Most bacteria are unable to grow in 7.5% sodium chloride, but there is one important exception. Staphylococcus species grow quite well in 7.5% sodium chloride. So mannitol salt agar is primarily used to select for or support the growth of Staphylococcus species. At the same time, it will select against or inhibit the growth of virtually all other species of bacteria, with a couple of exceptions. There are a couple of species of bacteria that can grow on MSA, but they can only grow quite poorly. And when I say poorly, I mean the colonies are going to be smaller than normal, and they're going to be poorly developed. These include Micrococcus species and Enterococcus species. They can grow on MSA, but they will only grow quite poorly on the medium. Staphylococcus, in comparison, will grow quite well on MSA. Now, MSA, it turns out, is also a differential medium, and we'll talk about that in just a minute. A good example of a differential medium is one called McConkey agar. McConkey agar is going to help, again, differentiate between closely related species or strains of bacteria. What McConkey agar can do is differentiate between bacteria that are able to ferment lactose from bacteria that are not. Now, there are lots of bacterial species in the human gastrointestinal tract that are able to ferment this sugar called lactose. Lactose is just milk sugar. When organisms ferment lactose, they create an acidic metabolite. The acidic metabolite is going to collect in the media, and it's going to lower the pH. That change in pH is going to cause the indicator dye in the McConkey agar to change color. Now, in this case, it's not the medium that will change color, but the colonies themselves. If the bacteria is able to ferment lactose that's in the agar, the colonies are going to grow with a pink color. Organisms that are not able to ferment lactose will not cause the production of this acidic metabolite, so the pH of the medium won't change, and the colonies will grow as colorless. Something else to note is this. Any type of bacteria that's capable of fermenting lactose gets a special name. We refer to those bacteria as coliforms. Now, McConkey agar is both a differential medium and a selective medium. We'll get to that in just a minute. So, we have two kinds of growth media, basal media and enriched media. We have two kinds of special media, selective and differential. There are two other large categories of media. One is called transport media, and one is called storage media. Transport media, like the name suggests, is designed for the transport of microorganisms from one location to another. Transport media provides just enough nutrients to support the survival of bacteria during transport. It provides for about 24 hours. Now, the important thing to know about transport media is that it is not designed to encourage growth. And if you think about how it's used, that makes sense. When a physician gathers a sample at the bedside of a patient and sends it off to the microbiology laboratory for analysis, the sample cannot simply be sent in any old container because the cells will die along the way. So instead, the sample is placed into some transport media. There are kits that physicians use at the bedside to do things like swab lacerations or swab the back of a person's throat. And those swabs are then placed into a small volume of transport media so they can be sent to the laboratory. There's just enough nutrition inside the transport media to keep those cells alive for about 24 hours. We don't want those cells to grow. We don't want them to multiply on the way to the laboratory, because then it will appear as though there were more cells in that patient's wound or in the back of that patient's throat than there really were. So transport media is designed to keep the cells alive, but not allow them to grow and divide. It's important, of course, that the companies that transport these samples between a physician's office or a hospital and the microbiology laboratory, it's important that they're able to deliver within about 24 hours. Because if they get out much past that, if you get to 36 or 48 hours, the transport media won't keep the cells alive. The last type of media on our list is storage media. Now, storage media is probably better referred to as freezer media because that's what it's used for. Storage media is used to keep a sample of microbial cells alive, but at very cold temperatures. Remember, part of the problem that we have with freezing living cells is that there's water inside of living cells, and water has the strange characteristic of expanding when it's frozen. The water inside of living cells will expand when freezing occurs and actually break apart the membrane of the cell. So when we try to thaw out the cell, we won't be successful. The cell will be dead. What storage media does is use a molecule called glycerol to replace the water inside of the living cells. When you place microbial cells into glycerol, the glycerol will go into the cells and force the water out. Glycerol won't harm the cells at all, but it will remove the water that can be so damaging upon freezing. When the glycerol-filled cells are then placed into the freezer, the cells can be stored for long, long times at the low temperature and then thawed out and survive the process. We often use storage media to freeze microbial cells at minus 80 degrees Celsius, which is a very cold temperature. We can keep microbial cells for an unlimited period of time, really. And when we thaw them, they'll still be viable. Now, just like transport media, storage media is not designed to encourage growth. We're just going to help keep the cells alive through the freezing process. Let's return now to the topic of special media. It turns out that several of the special media are both selective and differential. I mentioned a few minutes ago that mannitol salt agar is one of these. Mannitol salt agar, as we said, selects for Staphylococcus species and selects against most other kinds of bacteria, with a couple of exceptions. Mannitol salt agar is also differential, and it will help us differentiate between bacteria that are able to ferment mannitol from those that cannot. Mannitol is simply a sugar alcohol, and some bacteria are able to ferment it. When bacteria ferment mannitol, they produce, again, an acidic metabolite. That acidic metabolite will accumulate in the medium, cause the pH to go down, and then cause the indicator dye in the medium to change color. The type of indicator dye that's found in MSA is called phenol red. It starts off as a reddish pink color in the agar, and if the bacteria that grows performs this metabolic pathway and produces this acidic metabolite, the phenol red will turn yellow. So, Staphylococcus is the only type of bacteria that will grow well on MSA. And there's a certain species of Staphylococcus called Staph aureus that is a mannitol fermenter. When Staph aureus grows on MSA, the medium itself is going to turn a yellow color because Staphylococcus aureus ferments the mannitol inside the medium. Bacteria that can't ferment mannitol, other kinds of Staphylococcus, will grow on MSA, but they won't do that metabolic pathway. They won't ferment the mannitol. No acidic metabolite will be produced, so the pH stays the same, and the medium stays that pinkish color. Staphylococcus epidermidis is a good example of a mannitol non-fermenter. On this slide, over on the left, we can see an uninoculated MSA plate, mannitol salt agar plate. It has this pinkish color to it because the medium has phenol red indicator dye in it. Over here on the right, you can see that two types of bacteria have been streaked onto this plate. One of them is growing on a pink colored medium, and the other one has turned the medium yellow. This is Staphylococcus epidermidis. This is Staphylococcus aureus. We can look at an MSA plate that we've inoculated, and we can know that if something is growing on it, it's a Staphylococcus. I should say, if something is growing well on it, it's a Staphylococcus. And if what's growing on it has turned the medium yellow, we can say that it's a mannitol fermenter. Staphylococcus aureus is a mannitol fermenter. Staphylococcus epidermidis is not. Another medium that is both selective and differential is MacConkey agar. If you look at the top of the slide, you can see an uninoculated MacConkey agar plate. It has a very similar pinkish color to mannitol salt agar. We said a few minutes ago that MacConkey agar can differentiate between bacteria that can ferment lactose from bacteria that cannot. Remember, any bacterium that can ferment lactose is called a coliform. A good example of a coliform is Escherichia coli. E. coli is a gram-negative enteric coliform. That word enteric just means that it likes to grow in the gastrointestinal tract of mammals, including humans. E. coli is able to ferment lactose, and when it does, it produces an acidic metabolite that reduces the pH of the medium. The indicator dye in the medium doesn't change the medium color, but rather causes the colonies to change color to pink. If you look at the image over here on the lower portion of the slide, you can see some of these pink colonies growing on the MacConkey agar. Another good example is Salmonella enteritidis. This is one of the bad enteric pathogens in humans. Salmonella enteritidis is gram-negative and enteric, just like E. coli, but it's a non-fermenter. It is unable to ferment lactose. So when Salmonella grows on MacConkey, the colonies are colorless. MacConkey agar is also selective. MacConkey agar can select 4 gram-negative enteric bacteria. So if it's a gram-negative organism that lives in the gut, it will grow on MacConkey agar. It will select against gram-positive bacteria. So any organism that grows on MacConkey, we know, is a gram-negative type of bacterium. And if the organism grows with pink colonies, we also know that that gram-negative enteric bacterium can ferment lactose. Another example of a special medium that is both selective and differential is one called eosin methylene blue agar, or EMB. EMB selects 4 gram-negative enteric bacteria, just like MacConkey agar does. It selects against gram-positive bacteria, just like MacConkey agar does. What makes EMB and MacConkey agar different is what they differentiate between. Just like MacConkey, eosin methylene blue is going to help us identify lactose fermenters. But unlike MacConkey agar, EMB is going to help us differentiate a little more closely. Lactose fermenters, or coliforms, are going to produce those acidic metabolites that we've mentioned before. On EMB, the change in pH is going to turn the colonies a purple color. Remember, the colonies grow pink on MacConkey if they're lactose fermenters. They're going to grow purple on EMB if they're lactose fermenters. Now, here's where EMB goes even further. EMB can differentiate between a couple of common types of coliforms. When E. coli grows on EMB, E. coli is a lactose fermenter, a coliform. It will grow as purple colonies, but those purple colonies are going to have a very distinctive metallic green sheen on them. Now, that green metallic sheen is going to be best seen in areas with heavy growth. I'll show you what I mean in just a minute. Other types of coliforms are going to grow as purple colonies without the sheen. Any type of gram-negative enteric bacteria that is not a coliform is going to grow with no green sheen as colorless colonies. Remember, if it can't ferment lactose, it's not going to produce those acidic metabolites. There's not going to be any change in pH. So the colonies will grow colorless. On this slide, we can see the 3 ways that EMB can differentiate gram-negative enteric bacteria. Over here on the left, we can see a plate with growth on it You can see some streak lines here and some individual colonies. This was a quadrant streak plate. We know that if anything grows on EMB, it must be gram-negative and enteric. It must grow in the gastrointestinal tract normally. These colonies are also purple. That tells us that whatever this is, it's a gram-negative enteric coliform. It's able to ferment the lactose that's present in the media. In the middle image, we see a little something extra. We can also see this quadrant streak plate with isolated colonies. But notice that in the areas where the growth is heaviest, there's also a green metallic sheen present. This tells us that we have E. coli growing. Finally, on the last image, we're looking at an EMB plate with growth on it, so we know it's a gram-negative enteric type of bacteria. But the colonies are not purple. The colonies are colorless or even white. This is showing a lactose non-fermenter. In other words, it's not a coliform. It's gram-negative, it's growing in the intestine, but it is not able to ferment lactose. One more type of special media that we're going to talk about today is called Simmons citrate agar. Simmons citrate is usually produced as an agar slant. Because of what we're going to use it for, we don't need to see individual colonies growing. We just need to grow the microbe. Simmons citrate medium contains citrate or citric acid. Citrate is just an organic acid, a carbon-containing acid, and it's produced as a metabolic intermediate in a type of reaction called the Krebs cycle. The Krebs cycle, if you recall, is part of the process of cellular respiration. Now, Simmons citrate medium is both selective and differential. It's going to select for gram-negative bacteria that are able to use citrate. They're able to do something with citrate. And generally that means they're able to use citrate as a carbon source. They're also able to use ammonium as a nitrogen source, but don't worry too much about that detail. Focus on the citrate instead. Simmons citrate agar selects for gram-negative bacteria that can use citrate. It selects against gram-positive bacteria. So once again, we have a medium that's going to help us determine whether or not we have a gram-negative organism growing. Now, Simmons citrate agar is going to also differentiate between closely related species. What it's going to differentiate between is bacteria that produce an enzyme from those that don't. This enzyme is called citrate permease. Citrate permease is going to help during the process where citrate gets converted to a molecule called pyruvate. There are actually a couple of enzymes involved in that metabolic pathway, and citrate permease is one of them. So, if an organism has citrate permease, it can take the citrate in Simmons citrate agar and it can help to convert it to pyruvate. Pyruvate is just another metabolite that happens to be part of a couple of different metabolic pathways, including cellular respiration. Let's take a minute now and look at the mechanism by which Simmons citrate agar helps differentiate between bacteria that have citrate permease and those that don't. Remember, the process that converts citrate to pyruvate has more than one step, and more than one enzyme. The first thing that happens is the citrate molecule is broken apart by an enzyme called citrase. The citrase converts citrate to oxaloacetate and acetic acid. That's the first step of the reaction. The next step requires the enzyme citrate permease. The oxaloacetate and acetic acid will be converted to pyruvate and carbon dioxide, but only if citrate permease is produced. Organisms that lack citrate permease cannot convert citrate to pyruvate. Remember, pyruvate's just a metabolite, so once it's produced, it's going to be directed down some metabolic pathway. The carbon dioxide that's produced will mix with sodium and water inside of the microbial cell and produce a molecule called sodium carbonate. Now, this molecule is important because sodium carbonate is alkaline. This is where a pH change is going to occur, and the media is going to change color. Now, Simmons citrate agar contains an indicator dye called bromthymol blue, and under alkaline conditions, bromthymol blue is going to turn blue. Simmons citrate agar begins as a green medium. I'll show you in just a moment. And if the bacteria that grows on it produces citrate permease and converts the citrate to pyruvate and carbon dioxide, we're going to get sodium carbonate made, and the sodium carbonate is going to raise the pH of the medium. It's alkaline. That change in pH is going to cause the medium to go from green to blue. On this slide, over on the left, you're looking at some Simmons citrate agar in a slant. It's hard to tell that these are slant tubes because the agar slant is actually facing us. But just like all agar slants, we can streak bacterial cells onto the surface and then allow them to grow. This tube over here is what an uninoculated Simmons citrate agar slant looks like. It has this sort of deep, sort of forest green color. After incubation, if the bacterium growing on the slant contains citrate permease, we're going to see this happen. The medium is going to change color under alkaline conditions to this blue color. Now, Simmons citrate agar doesn't have to be used as a slant. You can also just use it in an agar plate. This plate was inoculated with two different types of bacteria, E. coli on the right, and an organism called Enterobacter aerogenes on the left. Now remember, Simmons citrate agar is both selective and differential. We said it selects for gram-negative enteric organisms, and it selects against gram-positive organisms. So if you get growth on Simmons citrate agar, you know you have a gram-negative organism. Both E. coli and Enterobacter aerogenes are gram-negative enteric bacteria. One of the ways that we can differentiate between them is by the presence or absence of this citrate permease enzyme. These are two closely related bacterium. They're both gram-negative. They both live in the gastrointestinal tract. But as you can see, E. coli does not have the citrate permease enzyme. It cannot convert citrate all the way to pyruvate. Enterobacter aerogenes, on the other hand, has that enzyme. It can take the citrate in the medium and it can help to convert it to pyruvate. Then the CO2 that's produced during that reaction will be converted into that alkaline metabolite, and the medium will turn blue.